In the past, when I've tweaked my ankle or had a headache, I've truly appreciated the quote unquote pure magic of ibuprofen. However, since reading this article from the Journal of Medicinal Chemistry titled Beyond Bioisomers, the Impact of Replacing a Benzene Ring, I've started to appreciate more the pure science behind ibuprofen and drugs similar to it. The Gen basic principle that shape determines function largely applies within the medicine world. As you'll see here, all five of these common drugs, including ibuprofen, share this yellow highlighted ring known as a phenyl group. The reason for phenyl group's popularity is because it's a benzene ring with one hydrogen removed, meaning that it has these alternating double bonds here, giving it two unique properties that we'll be looking at. It can have numerous pi bond interactions, one, and two, it has an openness to substituents being added. These two properties allow it to be more specifically targeted through sterics or changes in electron density to the ideal treatment area, along with optimization of physiochemical properties and the ADMET profile. The acronym I just mentioned stands for absorption, distribution, metabolism, excretion, and toxicity. This profile is five different categories in which drugs are assessed for their safety and efficiency. Typically, the incorporation of phenyl ring will help optimize this profile. However, in its over-incorporation, we start to see a decreased levels of ideal toxicity because of the formation of reactive intermediates. The danger of these reactive intermediates comes from the fact they're starting to interact with non-treatment areas, causing changes to proteins and macromolecules in undesired ways. So, past research has helped identify how these reactive metabolites or products from metabolism are actually occurring. We'll start off with that phenyl group, and you'll see it interacts with CYP450, which is an enzyme family common to the liver cells, and form an epoxide. This epoxide then goes under isomerization to form a phenol in ring opening. Further oxidation leads to dihydroxylbenzene. And then finally, the second reactive metabolite, quinones, are formed through further oxidation. The known presence of these reactive metabolites is from the formation of a GSH adduct, which is formed through direct nucleophilic addition or 1,4 conjugative addition. GSH stands for glutathione, which is a vital antioxidant in the body, usually part of a detoxification process in liver cells. The adducts themselves are a signal that this detoxification process has started, or them themselves can become reactive metabolites as they're further metabolized. We'll be looking mostly at GSH conjugates, which fit within the adduct larger category and mostly just signal the start of that detoxification process because of the presence of reactive metabolites. Past research has also shown that there's an ideal kind of hierarchy to the types of rings included within a pharmaceutical compound. Unfortunately, that benzene ring you'll see pictured here in the carboaromatic category is the least efficient, whereas the heteroaphylactic you'll see here has no conjugated or alternating double bonds and also includes non-just carbon-carbon bonds within its ring. You'll see oxygen pictured in this example. Frigid bicyclosystems fit within this last category. They've also been identified as saturated biastereoers of benzene rings, means that they'll have more sp3 rich character and they won't impact the function of the molecule if they're substituted for a benzene ring. Unfortunately, little is known exactly what happens once this switch is made to metabolic stability and the biotransformation differences. And therefore, we have this study. This study aimed to look at one, the difference in metabolic stability and other properties that come about when a phenyl group is changed to a more rigid bicyclic analog, to see what happens if to the metabolic fate when you add a substituent to this new analog, and then also what happens when there's not just carbon-carbon bonds. All these goals can be kind of summarized into one big question. Are bicyclic systems viable replacements for phenyl groups for drug production? So to see improvement, you first, of course, have to have a baseline. So that's where this article started. It created this molecule named 2C here. It has two benzene rings, as you'll notice, but only this one is highlighted in yellow because the one on the left won't actually be metabolized because of the steric hindrance of the fluorine substituents added purposely to more target where they're looking at in the molecule. It also includes an amide, as you'll see this functional group. And the importance of having that amide is it's so close to this benzene ring, it can be more easily removed once we want to create our bicyclo analogs for comparison later on. So to form 2C, they took 3,4,5 trifluoric benzoic acid and combined it with aniline in DMF solvent with a T3P dehydrating agent. Basically, they were creating an environment where it would be easier for nucleophilic attack from the nitrogen in the aniline to over here to the acid in the formation of 2C. Once 2C was formed, it was incubated in microsomes, which are small vesicles from the ER that basically emulate the environment of metabolism because it contains multiple important enzymes, such as that CYP450. From a 30-minute incubation, they saw the formation of two metabolites, one of which had that GSH tag to it. They also noted there's an oxidation with the O's and other conjugations with P. They took identification through mass spectrometry and retention time, which will be used later on to identify important steps. So since they know a reactive intermediate was there, they wanted to estimate how it was coming into being. So they used an anilinide, which has a similar structure to 2C with that phenyl group, as kind of a baseline because it had already been established a metabolic pathway. So they hypothesized that 2C would go through oxidation with our enzyme into an epoxide, which would be isomerized into a ring open, and then further oxidized with the removal of two hydrogens into 2I here, which would be that reactive intermediate as GSH was then added after. So to confirm if their pathway was correct, they kind of broke up the stages of what they were doing and formed an intermediate from their prediction from benzoic acid derivative and 4-aminophenol. And they took that intermediate and incubated in microsomes to see if they would still get their final product. So basically, they were looking at if each stage would actually lead to their prediction in the end of that metabolite. And they were indeed correct as they matched their new incubated from the intermediate to their original incubation of 2C in the beginning. So the metabolites matched. So now they have their baseline, they moved on to a method of matched molecular pair to create their bicyclic analogs, which means they're only going to have 1 point of specific structural difference between their old phenyl group and the new trying molecules. So you'll notice this yellow here gets exchanged for these difference in green on the right side of this diagram. The up top here are more bridged bicyclic systems. These are sp3 rich still, but not bridged. And these are just classic iostereors with still some sp2 character. So to reach aim 1, we're going to look at metabolic stability and the comparison of properties that happened in this research. Half-life is their measure for metabolic stability. So a longer half-life indicates the molecule stayed longer in the body and therefore would be more stable. They also look at membrane permeability, plasma protein binding, aqueous solubility, and C-log-P. C-log-P is a measurement of lipophilicity calculated from the structure of the molecule itself. It's typically desired in this research article that all of these properties would be increased from the original 2C molecule. So to see if these changes, they incubated the bicycloanalogs in the microsomes all the same and noted that there was an increase in all half-life, indicating a greater stability of the new molecules. There is more variation between all the other properties. For example, take the solubility here, some increased and some decreased. They mostly attributed this to the difference in sp3 character. As you'll see here, still there's a variation in structure and also differences in lipophilicity. So when they saw a difference in the properties, they also wanted to check the metabolites of their new analogs. So they took 3A, 3C, and 3D and all incubated them in microsomes once again. They noticed since 3A and 3C are connected to the bridgehead carbon here, that they only formed 1 or 2 metabolites. However, when it was just connected to a carbon within the larger ring, as in 3D, there was more variation in the final metabolites produced. As you'll see, there's 3 different types, indicating it's a less predictable pathway. They confirm this by, again, incubating it in now hepatocytes, which are just more complete environments with more enzymes and kind of emulating that metabolism that usually occurs in the body. This still showed that there's no GSH conjugates formed. They also look at their unbridged analogs and found similar no GSH conjugates, but they did notice that from their monocyclic system here, there was a greater liability and variance in their final metabolites, as it was more unpredictable, forming 4 different metabolites in the end. So overall, major takeaway, we avoided our reactive intermediates originally of 2C. So how is this coming about? They kind of used a past radical mechanism identified for cycloalkyl alternatives. And since the bicyclo analogs fit in this, they kind of attributed this change to this mechanism. This mechanism uses hydrogen abstraction and oxygen insertion to lead to a non-reactive alcohol compared to our original harmful epoxides and quinones. So then they moved on to aim 2 and to see what would happen to metabolic stability if they added substituents to the bridgeheads within their new bicyclo analogs. They use 2 different methods for this, and these are the methods that they also usually use to create their original analogs. It was method A, and we already identified that as that uses that T3P dehydrating agent. Method B uses different reagents, as you'll note in blue, but it creates that same environment that allows for nucleophilic attack and then therefore the formation of these molecules. Overall, there is that desired increase in metallic stability here and also a higher permeability of all these molecules. Originally, if you'll notice here, this is a bridged cyclopentane ring, whereas here they used more variation in the number of base rings, carbons. They saw overall that same increase in stability and wanted to specifically highlight that the fluorine had a very good increase in stability, whereas methane had a more liability, as you'll see here in 3I. It didn't have that same drastic increase. Their final aim was to look at heteroatoms if incorporated into the ring. And they noticed that indeed there was that same desired increase in stability once oxygen was incorporated. So why did we start? We were looking for a viable alternative to phenyl rings, and this research article proposed that bridged bicyclic analogs could do that. They wanted to improve the ADM profile. It was so important because they needed to avoid reactive intermediates that were causing undesired changes within the body. Their findings did support that once they changed out this phenyl group for bicyclo systems, they did find an increase in stability that was furthered when they added substituents to the bridgehead or when they added oxygen into the ring instead of just carbon-carbon bonds. So it seems possible, yes, we can enhance that ADME profile if we exchange out those phenyl rings. However, this finding needs to be a little bit diluted in its excitement because we only looked at one type of molecule. It was 2C that this research article used as a base. So to make sure these benefits can actually be extrapolated, there would be more research done. Also, you might have noted that I mostly reported just lists of changes and properties rather than explaining them why these occurred. This is because this research article mostly observed changes rather than trying to explain them. So further research would also need to take place to kind of see why these benefits actually occur. In this further research, they might want to add other research methods of looking at why a reactive intermediate was formed, as they only use one testing of GHS conjugates in this research article. I originally chose this article because I was interested in the key vocabulary that had shown up in other articles, such as sp3 rich and bridged bicyclic. I was really encouraged to see real-world application so early on in class. But I was solidified in my decision once I learned what ADME profile stands for, as the bridge of or branch of science that I'm most interested in is where the body interacts with the chemistry and how we're able to assist it through our science processes to help more the body function on its own. I was overall even more encouraged as I continued reading, seeing that there was principles from almost every chapter showing up in real-world application of this article. I did find small frustrations in trying to understand the difference in biological processes. Of course, that hasn't been my focus thus far in trying to understand in education. So the difference in audience for this research article, of course, just created gaps. I also had to do a lot of outside research to understand mechanisms, as they very commonly skipped over just listing reagents and saying it was a single step reaction. I think the struggle was definitely worth it, though, as I got to see how a molecule that I use in my daily life is being impacted by organic chemistry. Thank you so much for listening, and you should like and subscribe below. And I hope this presentation helps you understand a little bit more about what organic chemistry is doing for the medicine world.